your attention. Let me give you a brief agenda of the event. There are 4 segments. First, welcoming remarks from Professor Shopian Shah Jayaputra, Rector of Institute Teknologi Indonesia. Welcome remarks from Dr. Aryo Betajua Silvero, Head of Research for Environment and Clean Technology, BRIN. Second, we will be hearing a lecture from our keynote speakers,
Jelas suaranya.
Dr. Arifin, co-chair of Small Island Research and Development Working Group, IOC Westpac, and Professor Mitsuko Makino, from PICES Organization, University of Tokyo.
Kok jadi gemetaran gitu sih?
As moderator, he is a lecturer in the Department of Agro-Industrial Technology.
Suaranya nggak jelas.
The session will be presented by the guest speakers, Dr. Sheila Kemka from PICES Organization, Fisheries Research and Education Agency, and RH. Arif Rahman, Magister Bioscience and Oceanography, Professor Mark Elwell, PICES Organization, University of Professor Charles Berry, PICES Organization, University of Toronto, Scarborough, Canada. Professor Sundar Isakomar, researcher in Research Center for Environmental and Clean Technology, BRIN, and lecturer in the Department of Agro-Industrial Technology, Then you will have general discussions with the participants where you can ask questions to our experts. And the fourth is closing speech from Institute in the Magister of Science and the Department of Agro-Industrial Technology, Indonesia. Without any further ado, we would like to welcome Professor Shopian Shah to open the event.
Oke, From Rektor ya. Apakah suara saya jelas ya?
Jelas, Prof.
Pening speed ya. Oke. Bismillahirrahmanirrahim. The honorary my voice is okay, clear, everybody?
Clear.
Okay. Okay, begin. Okay. Bismillahirrahmanirrahim. The honorary head of the research center for environmental and clean technology of BRINS, Dr. Aryo Beta Jun Silvero, MSc, with his staff, Dr. Alexander Baikov, Physics Secretariat and Project Coordinator of Physics for FishPhyto, all speakers and moderator, Professor General Arifin, Co-Chair IOC Westpac Working Group on Shiran, Professor Mitsuko Makino, Physics, University of Tokyo, Japan, Dr. Rednet Abu Anwar, Abu hadir, Department of Agro-Industrial Technology of Indonesian Institute of Technology, Professor Mark Wells, Physics University, USA, Professor Charlie Trick, Physics from Toronto University of Canada, Dr. Sen Takimura, Physics from Japan, especially Professor Suhendra Kumar, Research Center for Environmental and Clean Technology of BRIN, and also is also Department of Agro-Industrial Technology of Indonesian Institute of Technology. The honorary also Hari Prahman of Research Center for
Kenapa tuh sampel yang
Oceanography.
nggak dapat sertifikat? Hah? Yang
Vice
apa, Bu?
Chancellor,
Dia ada tulisannya. Yang
Head
masuk
of
nggak ada.
Bureau and Center, ITI lecture,
Udah.
and students at the ITI, all participants of the general lecture with participate in this conference room and also at ITI. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Good morning and salam sejahtera buat kita semua. Swastiastu, Namo Buddhaya, salam kebajikan, salam, and good morning for everybody. Greeting to all of us. Praise to gratitude, let us pray to the presence of Allah Subhanahu wa Ta'ala. Because of His grace and guidance, we are given strength on earth so that we can gather of this general lecture at the ITI campus or also in the Zoom for this sharing webinar and sharing session on this. So all of us also from the ITI campus also or other the world in participant in this conference or in this sharing session. We would like to thank to the PICES, the North Pacific Marine Science Organization, delegation, and its experts, and the main marine science, also from Indonesia, who attend and will present for the lecture for this session. Thanks also especially to Brains, who was represented by Dr. Aryo. Terima kasih Pak Aryo, the head of Center for Environmental Research and Clean Technology, and this staff, as well as participants and invites who attended this event. Ladies and gentlemen, as a confirmed with the committee, the general lecture was held with the aim of the increasing knowledge and increasing the capacity of human resources in the field of marine science and technology for monitoring aquatic environment resources in coastal areas and small islands, which is very useful for broadening all of us insight in the field of knowledge and marine technology, especially for the students, researchers, lecturers, scientists from the ITI, also from BRIN and other scientific institutions, or other university in Indonesia, also for other institutions all over the world. I, as the counselor or rector of ITI, Indonesian Institute of Technology, Institute Teknologi Indonesia, fully support the various efforts to build synergy to improve the competence and quality of Indonesian human resources. For this reason, we, on behalf of the ITI, Indonesian Institute of Technology, we would like to thank to PICES especially for agreeing to come to give a general lecture at the ITI campus and for its other activities, which are very useful for all of us, as well as for public and community, not only in Indonesia, but also all over the world. In accordance with the vision, mission, and role of the ITI, we started the organization, which was founded in 1984 by Professor Dr. Ing B.J. Habibie,
Nggak apa-apa.
is also former the third president of Republic Indonesia, and also the Minister of Research and Technology in National Development. Namely now, we call technology-based technology-based international university. Through now is technology and innovation-based university to answer the challenges and of national and global development for current being. Now we are in Industrial Revolution 4.0 through Society 5.0. With this era and also through to Society 5.0, we are also very concerned about developing technology and increasing human resources capability as condition in spurring in the development and utilization of natural resources, including marine science, to support the acceleration of national development. We hope through this public lecture, the transfer of knowledge and technology can proceed in accordance with the vision and mission of all of us in the line with the PICES program. So hopefully it's make the sustainability development for our collaboration. We hope that of this collaboration will also benefit for all of us and contribute to development of science and technology globally, also in the international world, especially in Indonesia, in Asia, also for all over the world. Wish you a successful person in the general lecture, and we apologize if anything not really perfect for this event. We can improve for the next. Thanks again for all of us, for all of you, also to participate for this conference. Now we officially open for this sharing session about the general lecture, ITI, BRIN, and PICES, FishPhyto and marine environmental monitoring for disaster mitigation of harmful algal blooms, HAB, and singular fish positioning CFP, officially open. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you for your words, Professor. On behalf of ET, the next is welcome remarks from Dr. Aryo Betajua Silvero. The time is yours.
Okay, thank you. Can you hear my voice clearly?
Clear.
Very
Okay.
clear, Pak.
Okay, thank you. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning and warm greeting to all of us. The Honorable Rector of ITI, Professor Dr. Insinyur Sopian Sahjaya Putra, along with all of ITI leadership and staff, Dr. Alexander Baikov from PICES Secretariat and Project Coordinator of PICES for FishPhyto, all distinguished speaker and moderator, Professor Zainal Arifin, as the co-chair of IOC Westpac Working Group on CIRAD, Professor Mitsutaku Makino from PICES Aori University of Tokyo, Japan, Dr. Renat Abu Amar from the Department of Agro-Industrial Technology, ITI, Professor Mark Wells from PICES University of Maryland, USA, Professor Charlie Trick from PICES University of Toronto, Canada, and Dr. Shion Takemura from PICES, Japan, Professor Suhendar Sakumer from Research Center for Environmental and Clean Technology, BRIN, and Department of Agro-Industrial Technology, ITI, and then Mr. Arif Rahman from Research Center for Oceanography, BRIN, Vice Chancellors, heads of bureau and centers of ITI, lecturers and students, and all participants. First and foremost, let us offer our praise and gratitude to Allah for allowing us to gather today at the ITI campus for this important general lecture. This event brings together esteemed marine science experts from PICES and Indonesia, and we are very honored to be part of it. On behalf of BRIN, I would like to express our strong support for the collaborations between the Indonesian Institute of Technology, or ITI, and the North Pacific Marine Science Organization, or PICES, where this general lecture is not only a significant academic event, but also a meaningful step in the ongoing cooperation among ITI, PICES, and Research Center for Environmental and Clean Technology, BRIN. This collaboration is very crucial in advancing scientific knowledge and addressing pressing issues in marine science, particularly those related to environmental monitoring, and the mitigation of any marine hazards. As we know, as the largest archipelagic country in the world, Indonesia must continue to enhance its capacity in marine research and innovation also. And I hope that today's event inspires further collaboration that will strengthen our nation's human resource capabilities, especially among students, researchers, and academics from ITI and beyond. So, ladies and gentlemen, BRIN is committed to becoming a reliable, professional, and innovative national research and innovation institutions, filled with integrity and dedicated to supporting the President and Vice President in achieving the sovereign, independent, and character-driven of Indonesia. So in pursuit in these missions, BRIN aims to first generate scientific and technological breakthrough to enhance national productivity, competitiveness, environmental quality, and disaster and climate resilience. Second, develop excellent and competitive research and innovation human resources infrastructure and also utilizations. And the third, uphold good and transparent governance. So we believe that the partnership between BRIN, ITI, and PICES is aligned with these goals. And we do hope that this collaboration will be instrumental in the strengthening research quality and building infrastructure that support innovation and sustainability. Anyway, before concluding, I would like to express my sincere appreciation to ITI and PICES for the generous donation of the plankton analysis instruments, which called plankton scope, to BRIN. And this equipment will significantly support our ongoing research activities. I also extend my gratitude to the organizing committee for making this event possible. So we do hope also this general lecture will be fruitful for all of us. And thank you and best wishes for a successful sessions. Assalamualaikum warahmatullahi wabarakatuh. Thank you.
Thank you for your welcoming us, Professor, on behalf of BRIN. Now we will take the first photo sessions. To all participants, please open your camera and our staff will take the photo. Please open your camera. We will take the first photo session. Okay, the first slide. Okay, it's done. We may continue our agenda. That is the first lecture from Professor Zainal Arifin. If you are ready, the stage is yours.
It's my turn now.
Zainal Arifin, maybe you can share your materials.
Good morning, everyone. First of all, Honorable Professor Sofyan, Rector of Institute Teknologi Indonesia, Honorable Dr. Aryo, Director Research Center for Environment and Clean Technology, my international colleague from Japan, Prof. Makino from Tokyo Uni, and Dr. Sion from Japan also, and colleague from Canada and Portland, Professor Charles Stick from University of Toronto, and Professor Professor Maxwell from University of Maine.
Coba namanya, gimana namanya?
Hello? Can you hear my voice?
Yes, sir. Go ahead.
Okay, let me continue. Please allow me to thanks also to organizing committee for an excellent preparation, as well as to my colleague, Professor Suhendar, for his invitation. So, dear colleague, ladies and gentlemen, today I would like to share to all of you about historical establishment of a network of small island research and development working group, and how gradually develop research program to support regional small island development network. Please allow me to share my presentation. Okay, how to... So my presentation today is on small island research and development for island sustainability. Small island are characterized by small land area or small population size. Islands considered small when their population less than 1 million and their land area between 1,000 to 5,000 square kilometers, yes. This is international consensus at area of tourism industry, yeah. But within Indonesia itself, the small island considered when the size is less than 2,000 square kilometers. So what is the mission of the small island research development network? Or we call it SIRAD, yeah, a Small Island Research and Development. There are two at least. First, we would like to see to carry out a strategic approach to empower small island communities. And then second, balancing economic growth with environmental preservation. So the main goal of the Small Island Research and Development Working Group, first, we would like to understand why a small island is important. First, small island can be found across geographic area of member state of IOC, Intergovernmental Oceanography Commission, Western Pacific region, yeah, under UNESCO organization. Each island has a specific geological feature. As a result, they are individually have a distinct ecosystem and host unique biological community. Secondly, island development increased dramatically in the last 10, 20 years. We can see it at least within our country in Indonesia, yeah. Many coastal ecosystems associated with islands are under pressure due to human development activities, include tourism, aquaculture industry, fisheries industry, and extractive industry. You can see for tourism and aquaculture industry, we can find mostly in Western Indonesian region, while extractive industry and fishing industry can be found in eastern part of Indonesia. What is the impact of these human development activities? This is some of identification that our group identify. First, on issue on coastal erosion, pollution, habitat loss, and lack of fresh water, clean fresh water. Yeah. So the issue of global climate change also impacted on sea level rise. Yeah. And last but not least is issue on lack of alternative livelihood. We found mostly in many parts of the West region. So this is the goal of working group. First, the goal is of a small island research and development working group is to facilitate a development resilient, inclusive growth, and sustainable prosperity. So from this goal, how we develop the network. It seems cannot work this one, right? Oh, see. It's hanging. Let me check again. Hold on. I think it seems there is a problem with the
interpolasi teh kalau misalkan si data yang kita ambil itu bolong-bolong atau enggak fungsi bisa diinterpolasi ditambahin.
tidak bisa digeser ya.
Saya tanya lah
Let me check first. I think I have to change the. No need to have a transition, I think. From my talk, I have to delete first. So this is the form of, this is the form of a network. Yeah? So on the left side of your screen, CIRAT Regional Network consists of several university in the region as well as research center and international organization. So currently, there are at least 2 universities that involve. That is Tokyo University and University of Science Malaysia. On the left, on the right side, this first circle is on research center at regional institution. There is a Korean Institute of Oceanography, Science and Technology, and Institute of Research Center, Island Research Center in China, as well as Research Center for Oceanography from Indonesia, and Research Center for Human and Humanities, Social and Humanities from RIN. Within a national network, we also have several institutions, include National University, Ministry of Fisheries and Ocean, and Ministry of Environment. So we try to have interdisciplinary work to tackle both research issue as well as development, island development agenda.
Many benefits from this network, not only for tourism, but also.
So there are 4 objectives. First, fostering interdisciplinary research collaboration, and then enhancing community engagement approach. So we are also involving many national organizations that link with community both at national as well as at local level. Third, supporting evidence-based policy making for sustainable small island development. And lastly, strengthening regional cooperation among countries. Currently, we have 5 members: China, Japan, Korea, South Korea, Indonesia, and Malaysia. Oh, sorry. So this is the timeline. We have several activities in the last 6 years. First, we have activity in 2021, inviting several colleagues within Indonesia. And then in 2022, we have island studies focused on Pari Island on the north of Jakarta, as well as Weh Island,
National University of Jakarta, Professor Sun Hong, Director of Office of International Affairs, and also Professor Han Kyulin, Professor in the Marine and Fisheries Resources.
And then
also our other international
period of 2023 until 2024, we have international workshop, inviting several experts in the region, as well as we actively join International Island Forum during the last 2 years in 2023 and 2024. So at national level, we also have several activities. So we have cooperation with university as well as with national non-government organization. So this is the long-term plan for the next 4 or 5 years. from this year till 2030. Yes. So several research has been undergoing, mostly baseline understanding and situational analysis of a small island. Research activity mostly on mapping the socio-economic condition of the local communities and study of natural resources. That's including what Professor Asep Suhendar working on harmful algae bloom, yeah, in Lombok Island, and then survey on clean water supply, issue related with waste management practices, as well as climate change adaptation. Excuse me. So this is the plan we do, yeah, we will do in the next 5 years. Excuse me. So this is the last slide, hopefully. So in the next 2 years, actually, we focus on 3 agenda. Focus on economy aspect, aquaculture and tourism research, and then on issue environment relating to Ocean Clean Index, freshwater resources, and wastewater management. And third, on social aspect. So this is my colleague from Research Center for Social and Humanities. Yeah. Lastly, I would like to share several islands that will involve in the next 5 years. Yeah, from Japan there is Amami Island, led by Professor Saito from Tokyo University, and in Indonesia there are Gili Ketapang and Gili Matra. Gili Matra in Lombok was led by Professor Suhendar, and in South Korea there will be Jeju Island, Malaysia Langkawi, and from China will be Pingtandao Island. I think that's all my presentation, and I'm sorry for the presentation is not good, not very smoothly presented, yeah, because of a problem on my part, I think. Thank you very much, everyone. So I stop here now. Thank you.
Thank you so much for your insight, Alipis. Thank you so much for your insight, Professor Arifin. We'll move on to our second keynote speaker. We will move on to our second keynote speaker, Professor Winston Sakumakino. Let me check with him. Hello, Professor. Professor Mr. Taku Makino. Okay, can you unmute yourself?
Okay, good. Can you hear me?
Okay, we can hear you.
Okay, perfect. Thank you. Thank you very much. And Professor Shopiansha and Professor Adio of Green, also my respected friends, Professor Zainar and Professor Sender. It is my very honor to be here and to attend this general lecture and scientific meeting online on FishPhyto and marine environmental monitoring for disaster mitigation of HAB and CFP. Also, I would like to show my sincere gratitude and appreciation to all the organizers of this general lecture. Thank you very much. My name is Mitsutaku Makino from PICES, North Pacific Marine Science Organization. And my affiliation is the University of Tokyo and Sasakawa Peace Foundation. Okay, now please let me share my screen. Can I share my screen? Yes, I will try now. Okay, can you see my screen? Okay, good. Thank you. Thank you very much again for inviting me to this very important lecture. My talk is titled PICES Projects for Creating Observing Program for Sustainable Local Community in Indonesian Coastal Waters. FishGIS, Shigatera, and FishPhyto. So basically, my talk is the background and history of our scientific activities as the PICES project and our history of our collaboration with my dear Indonesian colleagues. Okay, let me start. Our project, it is called the PICES MAF project. MAF means Ministry of Agriculture, Forestry, and Fisheries of Japan. So our project is funded from the government of Japan. And the history of our PICES MAF project started back to 2007, almost 20 years, very long history. And the very first project funded by MAF Japan was Seafood Safety Project. This project was led by Vera Trainer and Mark Wells and Charlie Trick. And actually, Mark Wells and Charlie Trick are today participating to this lecture. And one of the most important, success of this very first project is Mr. Arif Rajan. He was the participant of this project as the student, and now he is a very important, researcher in Indonesia and very respected colleague of ours. So, yeah, this is the very beginning of our project. Our second project started 2012, and I and Dr. Ian Perry are the co-chair of this second project entitled Marine Ecosystem Health and Human Well-being. And then our third project, building capacity for coastal monitoring by local small-scale fishers, called FishGIS. This project was led by me and Professor Mark Wells. And our fourth project is Shigatera from 2020, led by me and Mark Wells. And the latest project was from 2023, FishPhyto, also led by Mark and McKinnon. All 5 projects were conducted based on the collaborations between Indonesian researchers and Japanese researchers and Korean and PICES researchers, including Japan, Korea, Russia, United States, and Canada, and China. So the relationships between Indonesia and PICES has been a very core, very important part of our activity. And this is our philosophy, our approach we adapted to all the projects. Transdisciplinary approach. It is co-design with local stakeholders, co-production of knowledge with local people, and co-delivery with local researchers and local government. So, we, as a researcher, would like to work together and discuss together and research together with local people. This is our basic philosophy. And it is very similar to the, I guess many of you have heard of UN Decade of Ocean Science. And the basic or the very core concept of UN Ocean Decade is also co-design and co-deliver with stakeholders. So collaboration with wide-ranging stakeholders in Indonesia is the most important, part of our approach. And now I would like to show you what we have done. One of the outputs of our projects is a mobile phone application. It is called FishGIS. And this application was co-designed with Indonesian people to be used by Indonesian local coastal fishers and local people. As you can see from these photos, we organized, many local workshops at the coastal communities in Indonesia with variety of people living in the coastal area of Indonesia. And we collected many opinions, many viewpoints, and many sense of value from local people. And also we collected the local needs for our project. And based on the results from these, dozens of meetings, we identified 5 monitoring items: coastal water quality, phytoplankton, mainly the harmful algal blooms, fish catch estimate, illegal, unreported, and unregulated fisheries, and floating litter or plastics. These 5 monitoring items using the mobile phone are identified and co-designed with local people. This is a very important part. Please let me show you the image of how you can use the FishGIS app. Later in this general lecture, Dr. Shion Takemura and Dr. Mark Wells and Dr. Charlie Trick are going to show you more detail, but this is just an image introduction. This is a fish photo took by the Indonesian local people using this FishGIS application, mobile phone application. You can see many photos of the local fish. And then the place location of the photo indicated in this map on your left side. These photos help us to understand fish diversity and important fishes for local community in Indonesia. And Dr. Shion Takemura will talk about it later, but you can use these photos for, more for analysis. For example, size distribution or species identification using AI. Also, if you collect the data about the color of the water, you can estimate the water clarity and the quality of the water using this application. Another outputs, very important outputs by these our projects are the MOU between our organizations. This is the chairman Iskandar of ITI, and below you can see the secretary of PICES, Dr. Sonia Batten. We signed MOU between Indonesia and PICES. And many of us participated to this ceremony, including Zainal-san and Sehendar-san and Shion-san and Mark and Charles. This was a very important and very memorable moment for us. And based on this MOU, we established the system of our, kind of collaboration, research collaboration. And Professor Sehendar plays vital, very important role in, establishing this organization. Also, we conducted field studies in Lombok. And I would like to show my sincere gratitude to all the organizations and all the friends in Lombok. It was very, insightful field trip, and we got a lot of important data. And Arif, my friend Arif, found many, good research, and he's going to talk about it later, I believe. So this is actually my last slide. Capacity building is the key. As I mentioned earlier, Mr. Arif Rachaman is our trainee of the very first project. And, our project is co-designed with our Indonesian friends and colleagues. This capacity buildings is going to, nurture the strong support from the communities, and all the trainees participated to our past training workshop or past symposiums. We met many fishermen from all over the Indonesia. They will be the teacher of the local fishermen. And I believe they are going to contribute to the sustainability of Indonesian coastal communities and coastal ecosystems. This is our outputs, and we are very proud of, being a colleague of all the fishermen and all the local people to this project. Thank you very much.
Okay, thank you for your input, Professor Martino. All right, we will start the scientific session. Dr. Abu Ahmad, I will ask you to introduce our guest speaker.
Thank you very much, Ms. Gita. We would like to continue our session today, but before continuing this session, photo session, maybe taking photo session, Prof. Suhindar, is okay? Photo session? Okay. Please, Mas Anai, to take photo session, Mas Anai. Okay. Okay. Thank you very much. So we would like to continue our session. We have 5 speakers for 2 hours. So we divided 5 speakers. That means every speaker 20 minutes. The first speaker is Dr. Shion Takemura from PICES, Fisheries Research and Education Agency, Japan, with the title Smartphone Application to Collect Coastal Fisheries and Environmental Information for Adaptation to Changes in Marine Environment. 20 minutes time is yours, Dr. Shion Takemura.
Can you hear me? Can you hear me?
Yes, clear.
Yes, clear.
Thank you very much for my introduction. First of all, I would like to thank to Professor Sender and ITI colleagues in Indonesia for providing this opportunity to attend today's wonderful general lecture. Hello, everyone. My name is Shion Takemura. I'm a researcher at the Japan Fisheries Research and Education Agency. So I'd like to share my screen. Can you see my screen?
Clear.
Yes.
Okay, thank you. So today I'm talking about our smartphone application. Already Makino-san introduced. So our application to collect coastal fisheries and environment information for adaptation to change in the marine environment towards sustainable fisheries management that have developed in PICES special project over 8 years. Before I introduce our application, I'd like to explain my motivation for developing application. So global heat water is a big issue around the world. Focused on the surround water, my country, Japan, and other PICES member countries. Water temperature are rising in the North Pacific Ocean around 40 degrees north latitude. Focused on Indonesia, temperature are also rising in the surround water of Indonesia, so around 0 degree latitude. Also, many research papers have reported that global heat water affects to fish communities. For example, Chung and Frelisher 2020 reported that the biomass and distribution of fish, not only pelagic fish, but also ground fish and salmon, on the east coast of the North Pacific Ocean are changing due to global heat water. What about the Indonesian water? Because fish are important resource for the coastal community in Indonesia. Also, In Indonesia, many small-scale fishers depend on fish for their life and their income. So in recent years, various projects in Indonesia are collecting fisheries data, but fisheries data is still limited. So challenges for adaptation to climate change in data-limited small-scale fisheries management are following 3 points. First one is to detect changes in the ocean ecosystems. Second is to share this information rapidly among stakeholders. Third, to use it for the decision-making on adaptation measures. So, monitoring activities are really important for the climate change adaptation. Recently, monitoring activities are conducted by not only the academia, such as researchers, but also community members. For example, Humber et al. 2017 reported that the community-based monitoring is a powerful approach to assess the small-scale shark fishery of Madagascar. Their activities found a significant decrease in the average size of the shark caught. In addition, in the past few decades, we have got new technologies for the supporting community-based monitoring. The rapid evolution of IT, for example, smartphone and AI, are expected to be a breakthrough in the solving the above mentioned 3 challenges. Here, I'd like to introduce 2 advanced projects. First is the eBird project. In this project, over hundreds of thousands of users have been reporting more than 100 million reports per year using web and smartphone app. Then, biodiversity information reported by volunteers is used for bird conservation research, like this. And second project is Manta Matcher. In this project, participants from around the globe reported photo of manta rays' ventral marking with information about where the photos were taken. These data were utilized in a study to clarify the number of individuals, migration patterns, and population structures of manta rays using AI technology. Now, community-based monitoring is a new academic field that is called citizen science. That is a transdisciplinary approach. So, our project has been developing a smartphone application, FishGS application, for collecting coastal fisheries and environmental information based on community-based monitoring since 2017. The FishGIS application allows local fishers to collect fish size distribution data from catch images and water quality parameters from ocean color image. Then to share the result with local stakeholders, and finally to facilitate the conservation and sustainable use of coastal marine ecosystem. FishGIS applications can be installed from Apple Store and Google Play now. If you are interested in our application, please try it from the following QR codes. In this my presentation, I'm going to show you research activities that utilizing FishPhyto. So this slide already Matino-san explained this slide. This is an example of FishPhyto collected by the FishGIS application. These photos tell us so many fish species live in Lombok. In addition, these fish are very, very important food for the local community. This slide shows previous analytical framework using fish photos before 2024, so old one. At the step 1, we take a photo, so report photo using FishGIS application. Work time for reporting photo is less than 1 minute per one photo. It is very easy. But at the step 2, work time for identifying fish from photo is about over 5 minutes per one photo. It is not difficult, but it takes time. Step 3 is very easy. We just run code for the measuring total length from photo. It takes few seconds. Finally, we get the fish size distribution data. But we have some issue for this step 2, identifying fish, these procedures. This slide shows present, very new one, analytical workflow using FishPhyto after 2025. Now, we got the new AI technology for analyzing fish photos. We just upload fish photo to the AI platform, AI technology for analyzing fish, AI, so Fisher AI platform. Then this AI automatically identifies fish and measure total length. It takes only few minutes. So we don't need to identify fish by hand now. This slide shows the comparison result between AI and human, me. The upper graph shows the AI results, and the lower graph shows the human results. The results of Fisher AI and human results show the same trend. Also, this slide shows errors by Fisheries AI. Errors by Fisheries AI are within plus minus 5% of the total length. So these results suggest that Fisheries AI is a very good solution for measuring total length. Therefore, I have conducted a preliminary analysis for monitoring seasonal change of size distribution for tuna species using Fisher AI. This slide shows my preliminary results. We have obtained total length data for tuna species. Most fish is long-tail tuna and little tuna, kawa. From photos that we captured at the fish market in Lombok Island in January and July 2023. we can find peaks of the distribution of total length for both species changed from January to July. So, Fisher's AI allowed to detect seasonal change of size distribution of fish in Lombok Island. Now, we got the new technologies such as smartphone application and AI for measuring total length from fish photos. Next step is to accumulate yearly and/or seasonally data and to perform length-based stock assessment. To perform length-based stock assessment is not difficult. If we accumulate a lot of fish photos and total length data using by Fish GIS application and Fisher's AI technologies. So, data collection is key for monitoring for adaptation to climate change in data-limited small-scale fisheries management. And then again, community-based monitoring is very good approach for the collecting data. So this is my last slide. So I would like to conclude. IT, such as smartphone and AI, are expected to be breakthrough in solving challenges in data-limited small-scale fisheries management under climate change. Fish GIS is a tool for monitoring ocean conditions for climate change adaptation. Fish images support to understand change of fish stocks. And water color image help to understand of change in the marine environment. Today, today I didn't explain this application, but later Professor Mark Wells introduced this application. So future challenge is not only collecting data, but also establishment of a mechanism for local stakeholders to utilize monitoring data for decision-making on adaptation measures. That's it. Thank you very much for your attention.
Thank you very much, Mr. Sion Pakimuran. Very nice explanation. You have already explained us about how mobile application can be crucial tool for fishermen and other stakeholders to gather data about fisheries and ocean condition. Thank you very much. So we would like to continue our session today. The next presentation will be delivered by Pak Arif Rahman, with the title From Reef to Risk, Ecological Insight and Distribution of Camber Discus and Associated Benthic Ciguateric Dinoflagellata in Indonesia. Pak Arif Rahman, Master Biological Science Research Center for Oceanographic, National Research and Innovation Agency. Thank you very much, Mas Yon. Time is with Pak Arif, 20 minutes.
Test. Can you hear my voice?
Okay.
Is it clear enough for my voice?
Little bit more loudly, maybe.
Hello, test. Is it clear now?
Okay. That's okay, clear.
Okay. I will try to speak a little bit loud and hopefully um okay, thank you very much for introducing me, ladies and gentlemen, and also thank you for Makino-san, Xiang-san, Professor Mark, Professor Swenda, Professor Jenal, and all other colleagues for arranging this general lectures. And also, I would like to thank to PICES project that have paving the path for me to be here with you all today. I'm honored to be here and happy to meet all of you and share some of my knowledge in this excellent occasion. Now, the talk that I will deliver today is about the Cambiodiscus and also associated benthic zygotendina flagellate in Indonesia. I'm Arif Rahman, currently researcher in the Research Center for Oceanography and also a PhD student in the University of Montpellier in the Marbec unit of France. So a little bit introduction about my talk. We all know it has been repeated many times that the Indonesian coral reefs is the rainforest of the oceans and the hotspot for the marine biodiversity. And our coral triangle, our region is part of the coral triangles, which has more than 500 species of coral, and we have 3 centers of endemism inside our country. And so far, we know that the highest diversity of coral reef species was found, especially in the Birkhead Peninsula of Papua. We have more than 500 species, and with the highest coral reef species density in the world. And also, our coral triangle is also the hotspot zone for reef biodiversity, and our ocean is also the world's richest country of fish biodiversity, with more than 2,000 species per kilometer square. But our coral reef, if you all have known, is under constant threat, and we can split the threat into 2 types of threats. The first one is the acute threats that cause significant damage over a very short period of time. For example, a destructive practices for fishing, sometimes anchor damage of ship groundings, or natural disasters like cyclone, storm, and tsunami. And also we have problems with the chronic threats that changing the physical and biological components of the ecosystem and cause long-term damage. For example, the pollutions, uh, including the industrial and sewage and eutrophication, sedimentation, and of course, the global change that we see, the ocean acidification and ocean warming. Now, this is a figures that showing the ecosystem services that coral reef gives to human and to the general ecosystem in the coastal area, and also the threats that are faced by the coral reef itself. The threats that are faced by coral reefs, and this causes coral reef degradation, which of course it will reduce causing loss of key ecosystem services, including and will end up losing the biodiversity because the loss of biodiversity, and also will increase the case of disease, including in human. And I'm going to talk a little bit more about this kind of disease, which is the ciguatera fish poisoning, which has become the interest of our PICES project for the last 5 years. Now I'm going to talk about the Ciguatera fish poisoning. What we know about the Ciguatera fish poisoning. This is one of the most reported seafood-borne and harmful algal blooms-related disease in the tropical countries that is emerging. It is an emerging disease with expanding global distribution. There's approximately between 10,000 to 50,000 CFP cases per year, and WHO estimated that the number of people that get affected by the CFP could be much higher, between 2 million to 10 million people per year. This is a huge number. And this disease caused by ingestion or when we eat the seawater toxin contaminated reef fish, which cause many symptoms, usually within 12 hours of ingesting the contaminated fish. This is a complex disease because it has so many symptoms that are difficult to identify. Yes, like I said, this CFP is a complex disease. Currently, there are at least 175 symptoms that have been reported. Some are very short term, some are very long term. It could last for days, could last for years in some rare cases. So this is a very difficult to diagnose properly, and we have not been able to actually pinpointing the cause of such symptoms because this is a very complex disease. Like in COVID, in some cases, with we call it the disease with the 1,000 faces. So this is the chemical structures of the ciguatoxin. It is very heat stable because it has long chain. It has a polyaromatic cyclic. This is making the structure itself very stable. This is lipid soluble. It's dissolved in the lipid, in the fat of the fish, and it's very heat stable. So if you cook the fish, it did not destroy the toxin, unfortunately. And it's odorless and tasteless, so organoleptic test is useless. So you cannot taste the toxin, and so far there's no antidotes. So this is how the ciguatoxin can enter the human body through there's an environmental and an anthropogenic cases that cause the reef degradation, leading to gambidiscus blooms, and then they are starting to produce the ciguatoxin, which enters the food chain, food webs via the predation. And when the human eats the fish that are contaminated, we become sick. So this is another way to see the mechanism of ciguatera outbreak because of the coral reef degradation. If we have the acute or the chronic threats to the to the coral reef that cause the degradations or massive death of the corals that will quickly colonized by the macroalgae. Macroalgae promotes the growth of the benthic dinoflagellate and causing ciguatera. And at the same time, if we have a problem of overfishing, the predators that usually control the population of the benthic dinoflagellate are gone, and now we have the problem of the ciguatera in the coastal ecosystems. Now, the ciguatoxins is like a lot of toxins in the nature undergo process bioaccumulation and biomagnification, and sometimes even transformation. This is why it's very difficult to study the toxin itself. So as you go higher in the trophic levels, the risk to get intoxicated by the ciguatoxins also increase. So the carnivorous fish and the top predators usually is the high risk to accumulate high concentration because of the process of bioaccumulation and biomagnification. But in some cases, the herbivore that are grazing directly on the macroalgae on the coral also become a risk because they can accumulate a lot of ciguatoxin in their body. So now we're going to move to the cause of the ciguatera fish poisoning, which is I'm going to talk a little bit more about Gambia discuss later. Now, ciguateric benthic dinoflagellate is the one that causing the ciguatera fish poisoning in human. Actually, there's 5 that are currently the targeted genus of the ciguatera fish poisoning. Actually, there's 6, including a new genus, Fukuyoa. but I did not show it in here. But they are capable of producing toxic substances, mainly the ciguatoxin and some analogous toxins that are similar with the ciguatoxin and probably works the same way as ciguatoxin. And they are the group that we usually call as a benthic harmful algae or BHA. And then the ciguatoxin was first actually discovered from this particular species, the Gambia toxicus, from the Gambia island in France around 1980s or 1970s. So I'm going to talk more about the Gambia discus because this is the current topic of my research. This is one of the main cause of CFP. They mainly produce two types of toxin, but later I will show you they're producing a lot more metabolites, the secondary metabolite that also work similar way with the ciguatoxin, and they naturally attach to the surface of the many hard surface that you can find in the oceans, aquifers, corals, and drains, and even in the man-made surface and floating debris like plastic, woods, and etc. As you can see here, they are very big. If you see it, the cells under the stereo microscope, when they are blooming, you can see it very clearly. This is the cells. If attached to the red algae here in the natural substrate, the macroalgae, and here in the artificial substrate, which is the screen that was used to study the benthic dinoflagellates. Currently, we are trying to use a standardized methods to study the benthic dinoflagellate. They are commonly found in the warmer oceans like Indian Ocean, Caribbean, and tropical belt of Pacific Ocean in Pacific Islands. But now we found them expanding to the subtropical waters of Australia, Japan, and Mediterranean, especially because of the global warming that Xiong-san already mentioned in his presentation before. Now, a little bit about the diversity. There's many species of the Gambia discus. I just show you some of them and how briefly how to identify some of the species. Basically, to identify the Gambia discus, you have to see the plates. So benthic dinoflagellates, they have armor. This one is what we call the armor dinoflagellate. They have a thecal plate that you can actually see under the microscopes. This is best to see under the epifluorescence microscope with the carpofluor staining, for example. And the formula of the plates, the number, the shape, and the configuration of the plates are very important to be able to correctly identify the species, because this is important because not all Gambia discus produce toxin, but the one that produce toxins usually are abundant and they are the one that are very dangerous. Now, I told you that the Gambia discus produce a lot of toxins, mainly the ciguatoxin, and then later on we found another type of toxin. It's called the mycotoxin. Also, they also produce some others analogous toxins that has very similar structures, and we think it probably works similar way with ciguatoxin. This might also be the cause why the fish poisoning is so complex. The symptoms are so complex because if the causative organism produce a lot of toxin at the same time, it might cause multiple symptoms, while the other species could only produce one toxin that only cause light symptoms in human, and the other one produce multiple toxin and cause like more fatal or more heavy symptoms in human. So this is also another challenging aspect of the studying the ciguatera fish poisoning can be discussed and the toxin they produce. Now, a little bit insight about the relationship between Gambia discus and some key environmental factors. Here, I show you the temperature, salinity, and how irradiance reacted with the interacted with the Gambia discus. As I said, Gambia discus, they occupy wide range of habitat type and expanding because their ability to tolerance many environmental conditions. They are mostly warm water species, but some species can grow with the water temperature as low as about 15 to 19 degrees, but usually the optimal range is between 19 to 31 degrees, so it's a tropical species, basically. For salinity, they are less sensitive to salinity variation. They are not very limited by salinity. Some of them can grow in brackish water. Uh, Although it's very low, their growth is not optimal. They can survive in the brackish water environment. But generally, they prefer the high and stable salinity between 28 to 35, and usually the oceanic salinity between 34 and even higher at the hypersaline condition like 38, 40. Uh, they can still sustain the growth for many, and some, not all, but some Gambia species. They are also sweat tolerant, so even if the water is turbid, they can survive. And then also they survive the very high irradiance, meaning that they can even colonize the area in the intertidal zone where the sunlight is very, very strong. Although it limited their growth, many of them can still survive. And because of their acid tolerant ability, there's a research that find some Gambia discus can even survive at the 150 meters depth. They are not thriving, they are not growing well, but they can survive at that depth. Now, the other things that are interesting is about toxin production of the Gambia discus. Later on, in some of the research, we found that some researchers found that the increasing nutrients did not affect the Gambia discus abundance. So they are more limited to other factors other than the inorganic nutrients in the water. So the growth restriction of natural Gambia discus caused by another things. Some of the research suggested that the dissolved inorganic carbon also very important for the growth and the abundance of benthic dinoflagellate, like the Gambia discus, and also especially the interaction with host substrates because they are attached to the other substrate like macrophyte, like seagrass or macroalgae. So if the macroalgae or seagrass produce another bioactive compound and secondary metabolites, some can inhibit the growth of Gambia discus, some can promote and increase the growth rate of the Gambia discus. So it's also very important between host and the Gambia discus relationship. And also the process that disturb the boundary layers like currents, wave, wind can also affecting the abundance. The interesting thing is that with researchers, many researchers have suggested that the toxin production of the Gambia discus generally they are occurring during the higher NP ratio or during the P limited phosphorus limited condition. So there's an idea of the eutrophication or the enrichment of the nitrogen, a lot of nitrogen in the water that will increase the NP ratio, which later on causing the P limited condition could actually be the reason why some of them the discuss produce a lot of toxin. And we see in our research that the Gambia discuss they are yes they are more abundant and they like to be in the warm temperature at the higher NP ratio. The other things that are interesting that many people have tried to study is the interaction between the Gambia discus and the bacteria in the phycosphere area of their cells. So the marine bacteria, we all know that they are exhibit both positive and negative effects toward Gambia discus growth and toxin productions. The bacteria can help converting the dissolved organic matter and inorganic matter. At the same time, the Gambia discus produce some excess that will be helpful for the bacteria, so they are symbiotic and mutualistic, symbiotic with them. And also they also some bacteria also can produce vitamins and trace elements that will help the growth of the Gambia discus. At the same time, they also act as a disease for the Gambia discus. It also naturally control their population by some bacteria produce toxic substance or other bioactive substance that can inhibit the photosynthetic or metabolic activities of the Gambia discus, and also they can act as the nutrient competitors in the environment. So this is something some others very interesting research topic that could be pursued because not many information available. There are some researchers that are doing a very excellent job on this, but there are many other factors that we still don't know about the relationship between the bacteria and in other cases virus, for example, with the Gambia discus and how they can control the population of Gambia discus naturally in their habitat. Now we're talking
1
about
minute
the
more, Arif.
right. So it's a little bit long. I will try to skip a little bit. So this is how the 22 non-Gambia discus and the other species Pukuyoa that are recently split from the Gambia discus genus are distributed in the world. We are in the Southeast Asia. Later on, you can see my slide that will be posted later. What kind of species that has been identified in the Southeast Asia and East Asia region, including our nearby neighbor, Australia. And this is where the sugar toxin has been identified and ciguatera-fish poisoning has been reported. As you see, it's a tropical disease, but as you see here, now it's creeping up, expanding to the temperate region. For example, in Azores Island, in some cases, we have a lot of cases where epidemic of Germany in 2012 to 2015 that was caused by ciguatera-fish that was imported to the countries. But as you can see, they are creeping up to the northern and the southern area, like in South Korea and Japan. I think there's some cases and some research has already been done to identify the ciguatera-fish poisoning there, and also it's becoming a problem in the Australia, Eastern Australia, but we don't have any information about the Western Australia. Now, this is how the Gambia discus population has been cataloged and split into 6 different regions. You can examine this map later. But we are Indonesia in the region of C, which is belong to the East China Sea, Korean Strait, Gulf of Thailand, and Celebes Sea. We have specific types of Gambia discus species that have been identified and studied in this particular area. Now, this is because of banded dana flagellate associated with CFP in Indonesia. Unfortunately, we don't have many research that are conducted about this particular organism and particular disease. And psychotherapist poisoning especially was never fully formally reported or officially reported in our country. And research are very limited to the western area of Indonesia. This is like a conceptual map highlighting some key locations that I managed to gather from the information. Was compiled by our college, Ms. Riani Widyati, which is the only expert of the banded dana flagellate in Indonesia from University of Indonesia, my college. And I tried to reorganize them, and I found that there's another papers that are identifying the Gambia discus from Manado, so there will be another one point here. But we have practically no idea, no data on Kalimantan or Borneo, Maluku, NTT, and then Papua. Another thing about the Gambia fish poison is their jam dispersal. Because the export of import of the fish, some fishes, unfortunately, from Indonesia, Vietnam, and India has caused a problem in Europe. In Germany, there is an outbreak, epidemic of fish poisoning from 2012 to 2015 because of the fish that are imported from Vietnam, Indonesia, and India. Our fish causing a problem in Germany in 2014, unfortunately. So how to mitigate CFP in Indonesia? This is very complex. You can see this slide later, but this is how the FAO in 2018 suggests the pathway.
Kenapa kagak nyambung si Alfinya?
The pathway of Sigotera response model based on the meetings or based on the findings are very complex. And it will become a safety issues in Indonesia, especially if we wanted to export fish to other countries. We have to definitely make sure that it's free from Zigotera, especially if our export is reef fish. So it's complex, always complex in our country. So, but I believe that there's three components that has to be operate in union. We cannot move just one gear. We have to move all of the gear at the same time. We have to do research. This is our part that we are doing a research, and with the help of PICES, we are also trying to do the community action. We foster the promote the scale up the monitoring and public education and awareness campaign. This is the one that we are focusing mostly in the PICES project, and also we try to develop the strategies like Xiong-san already mentioned about the fish GIS, and later on I think Professor Mark will be explaining about the hydrocolor. That is some technique to rapidly detect the presence of not cytotoxin perhaps, but for the causative organisms and the changing in the environment that might help to predict if we will have a problem of Ciguatera in the area or not. And this is what we already done with the Ciguatera project 2, which is a continuation from the Ciguatera Indonesia that was finished in 2014. And then now we are continuing the FishPhyto. We're not sitting around. We are trying to establish and trying to do a lot of things and try to combine many tools that are available in our disposal to try to have the monitoring system of the harmful algal blooms, not only for Ciguatera, but also all the harmful algal blooms by promoting the citizen scientists in the coastal communities. In summary, Ciguatera is a hidden but increasing threat to national food safety in Indonesia because of coral reef degradation. And the main cause of the CFP, can be discussed, are very tolerant. Their range is expanding. Their occurrence started to be noted or reported in many areas in Southeast Asia, including and in other temperate countries. And the CFP mitigation is complex, but the key is that we need a collaborative effort because we cannot develop the mitigation strategy alone, but we have to do a collaborative effort to develop the mitigation strategy for CFP and other harmful algal blooms. I think that's all from my presentation today. Thank you very much. I'm honored to be here. My apologies if the talk is a little bit longer. Thank you very much.
Thank you very much, Pak Arif. So, nice presentation about Ciguatera fish poisoning. It's a type food poisoning caused by eating tropical reef fish that are contaminated with ciguatoxin. So we have to be careful eating from fish. The next session will be delivered by Professor Mark Well from PICES University of Maine, USA, with the title Fish GIS and Plankton Scope for Monitoring Marine Resources and Environment. 20 minutes, time is yours, please.
Okay, can you hear me okay?
Clear enough.
Yes, very good. Okay, and I presume my screen has come through. Is it okay?
Yes, clear.
Very good. Thank you. Well, I want to first begin by thanking the organizers, in particular, for inviting me to participate in this important general lecture series. And a hello to all my friends and colleagues there in Indonesia. I wish we could all be present in person. I am going to present to you information about one aspect of the project. Primarily, most of what I'll talk about is about planktoscope. But as my colleague Xiong-san brought out, I am also going to talk a little bit about hydrocolor. So if I can start here, let's see now, I'm supposed to, can you see my cursor? I'm circling.
Yes.
Yes, okay, good. I don't know how to make the cursor in that sort of nice way here. At any rate, okay. The primary, if you want to think about ocean and productivity and good fisheries, you've got to start at the base of the food web. And the base of the food web are the phytoplankton. The phytoplankton and the benthic plankton that photosynthesize. And these are what take the sunlight energy and create it a chemical energy that then is passed on up through the food web. And so one of the primary factors that we are interested in when we're thinking about ocean and productivity is the amount of phytoplankton. And we can estimate the amount of phytoplankton by measuring the amount of chlorophyll, because all phytoplankton have chlorophyll A. And shown here is an annual mean distribution on Earth of chlorophyll in the surface waters, where the greener parts are where there is more phytoplankton, and the bluer parts are where there is not as much phytoplankton. And so this is a special sort of part of the light spectrum. We know that your waters off of Indonesia in many areas are extremely clear, but you can see when you look in here that annually you do have quite a bit of green because you're close to the shore, which means there's nutrients coming in, which can help sustain the phytoplankton. And generally, the higher the biomass, the higher the production. And these are the grasses of the sea. It can go against you if you start having too much nutrient coming in and you have too much phytoplankton. Then you can generate other problems, including harmful bloom type problems. And so measuring chlorophyll is an important component of trying to understand what the quality of your water is. So with hydrocolor, which is connected in this FishGIS app that Dr. Takamura presented, it is uses the same approach as satellites for measuring the chlorophyll. So in that previous slide, all those chlorophyll data came from satellite measurements. Well, the hydrocolor app uses exactly the same type of approach, where here you take a picture with your iPhone. You have to take a picture of a gray card. And that's what's shown over here is this gray card here. And what that does is it helps calibrate the camera to what the light conditions are that day. Then you take a picture of the sky, okay? And that's the picture here of the sky, which tells us what the incoming radiation is like, incoming sunlight, okay? And then finally, you take a picture of the water, and with those three components, you get data that are used to calculate chlorophyll. Now, this is another image of this showing it how it can be different depending upon what the day is like. So here on the top, we have our gray card. We take the picture of it. We have a blue sky, and then we take a picture of the water. But if on another day, if we have a gray sky, okay, cloud cover, then the camera has to adjust for that. And that's why you use the gray card itself. It's a way of calibrating the lens on the camera to know what the wavelengths of light that are coming in. And from that, you can correct what the wavelengths of light coming out of the water are on that day. And then those data, Professor McKino presented this slide here. Those data give us measurements in the light spectrum of red, green, and blue. And because red, green, and blue, that's what the cameras on the iPhone use to make pictures, is a combination of the wavelengths at those, the relative amount of light at those wavelengths. So this does not give you directly chlorophyll, but by using this app, you will collect data, the red, green, and blue data, which then are uploaded to a server in Indonesia, which can be converted to chlorophyll once you have that, using the same approach as with the satellite data. But this has to be calibrated for your coastal waters. And a reef is working with that along with Professor Suhendahar to get those corrections so that they can take the data that are collected and give you the amount of chlorophyll in those waters, okay? So that's one aspect of the FishGIS app that will help understand the quality of water by understanding the relative amount of the plankton in the water, the relative amount of food that's available for higher trophic levels. So what I also want to talk about, though, is just because you know the amount of chlorophyll there, that's not enough. You really want to know something about who is there, who are the phytoplankton that are present there. Reef just gave a very nice presentation about one group of those phytoplankton, Gambreadiscus, that can be problems. But there are other species, or the mixture of species become very important for understanding what the quality of the water is like. It's also a very important measure to give you a sense of how the water quality may be changing over time, because as the water quality changes, you will see different communities of phytoplankton emerging. And so these data are very useful to be able to understand that system. What also is very important, as Arif pointed out, is you want to know when there are particularly problematic species that are appearing. So you have to have a way of doing this. Now, what the ones that Arif presented is ciguatera fish poisoning, the Gambrinidiscus group of species. And I'm not going to go into detail here because Arif gave a very nice summary of what it is. This is not the only problem species. This is the one that our project has been focusing on because we had to choose one. But there are other harmful species that produce toxins that also you want to be able to look out for, because when you see them, then you know that, okay, we need to take steps in order to protect the humans that may be consuming different components of the food web. Now, with ciguatera fish poisoning, as Arif pointed out, at this point, we're not sure that it's present in Indonesia, but learning from other island nations that have gone through a lot of development, what has been found is that as you get more and more development, human development on shore, you often will see an appearance of ciguatera fish poisoning. So there is an association between the developments on the shores and the potential for Gambrinidiscus or other. Gambrinidiscus is one of the main ones that we're worried about with ciguatera fish poisoning. And so as we've heard, Indonesia is having a lot of more development in their small island areas and in large island areas. And it's that development, the concern is that while there is not a big amount of ciguatera fish poisoning, it would seem, in Indonesia right now, you want to be watching for this, because if you start seeing an increase in the abundance of Gambrinidiscus, you may be able to take steps to prevent it from becoming really well established in your coastal waters. And so being aware of it or looking for it ahead of the problem can be a tremendous benefit in trying to make sure that you don't get the same type of environments as you find in other islands, where communities don't eat the fish because they're too toxic. And you don't want to obviously have that happening in Indonesia. So the point is, we want a way of looking for this type of species and other harmful algal species. But as Arif can testify, it takes a lot of effort to do this manually with a microscope. You have to collect the water, you have to filter it, you have to then put it under a microscope, look throughout the whole slide, count cells, look for particular. It takes a long time, and it takes a lot of expertise to be able to do that sort of identification. So ideally, what we want to try and do is automate this in order to be able to allow a lot more samples to be collected more quickly. So planktoscope is basically an automated microscope, okay? It takes images of all the phytoplankton in the water, all the plankton, including zooplankton, in the water sample, and it can automatically count each type of those, okay? It doesn't do the identification directly, but you can see it'll know this looks the same as this looks the same as those. I can now say I've got 3 of those, even though I don't know what it is, right? We'll get to how you figure out what it is in a moment. So there are, you can build these, you can either purchase them from this company that is producing them and selling them at a reasonable rate for an instrument, Ferroscope, or you can, it's an open source, so the plans for building it are available. And so for about $800 US, you can actually build one of these yourself. So it is if you want a poor man's instrument, because sorry, I thought I had another, later on I'll say, but I mean, you can buy instruments that do this, that are tens of thousands of US, and they're very expensive. And so there's not many of those. And obviously that becomes a problem if you are trying to take these instruments around and look at different locations. And so this provides a low-cost approach to trying to do this. So it's been produced from a lab. The Manu Prakash is a professor at Stanford. This is a group of his students from about a couple of years ago now. And he was the one that basically got his students to start developing these low-cost instruments to allow them to be used in nations that can't necessarily afford expensive equipments, but also allow other people, citizen science, to get involved if they wished. This is an old slide now. This is over 30 different replications of that instrument. These are all built based on the open source information about how to do this. They can be made out of wood, they can be made out of plastic. People have taken different approaches to doing this, but it is available for you to do, or as I say, it's available if you want to purchase one. Planktoscope has a global user community. Shown on the dots here, there's different dots, different color dots depending upon how people are using those. But you'll notice that there is some dots here in Indonesia because Planktoscope has been part of some of the capacity building workshops that we've had as part of this project. And both Arif and Professor Suhendahar are experienced in these and are the people to be getting in touch with if you're interested in trying to either build a planktoscope or certainly to try and use it in providing information for Indonesian coastal waters. So how does it work? It's a beautiful, simple instrument in that you have here, the source of water. Now, often what you'll do is to enhance your ability to do this, you will use a plankton net to concentrate a certain amount of phytoplankton from the water, but then you just simply pour that water into this, it's literally a syringe, and from there it is pumped through to a imaging chamber, and it just flows through the imaging chamber and then back up into a waste tube, okay? And so as it goes through, it's a stop flow. So it'll pump and then stop, and then a picture will be taken. And then it'll pump and it'll stop and a picture will be taken. And it collects data quite quickly as you go through here. You've got the lenses here and the camera. You have a computer that then works to collect that image and to basically compartmentalize it. And as I'll show you in a second, and you also have a GPS on this so that it knows its location on Earth. So you can take this instrument and go to different parts of the coastal region. You can then do a net haul, take the plankton, do the counting. It knows where it is, so it collects that information. And all of this is hooked up through Wi-Fi or cellular. And those data can be transplanted back to the main server in order to be able to collect and work with those data. The images it creates are amazing. For the cost of the instrument, they're phenomenal. And Arif can talk to how good the type of images are with this compared to very expensive microscopes. And so you have the potential of doing really high resolution type work to get a very good sense of what the species are. These images came from a cruise that ran down through the Pacific, and the planktoscope was used on that cruise. And these are collected by scientists, but this can be done by with very little training, you can operate this and get this quality of images. So how exactly does it work with these data? So you've got here on the top up in here, we've got different pictures that were taken. These include zooplankton and also with phytoplankton. That is a Catossaurus, a type of diatom here. That's a dinoflagellate here. And what it does is it takes the raw images and it goes through a program that segments that raw image into a bunch of little images of the things that occurred in that big picture. Okay, so as that water pumps through the planktoscope, stops, an image is taken, it will have a number of different things in that image. That image then is taken to the processor in the planktoscope. It goes through the segmentation, and these are the images that end up being sent through the cellular to whatever your receiver or to the server which you're sending it to. And this way, it reduces the amount of data transfer. You're not sending all of these very high resolution, high data images. You're just sending the pictures of things that are important, okay? And those images then are uploaded to Ecotaxa. Ecotaxa, and this is what's being used right now for most of the global users. Ecotaxa is a database that allows you to compare images with known phytoplankton species or zooplankton species. And so this is an example here where you've got, in this case, a dinoflagellate that you're looking at here, okay? This is a serratium species. These images are what's been uploaded in the Ecotaxa. All these images are taken at different orientations. And so that image which is collected with the planktoscope will be compared through image analysis with all of these images. This happens to be just one species, but there are hundreds of species, thousands of species in Ecotaxa in order to compare that image to help identify it. Now, where I won't say problem, but where one of the issues is right now is that these are uploaded by basically scientists, users. And so what's needed is those type of images being uploaded from Indonesian waters. In other words, the species of phytoplankton in Indonesian waters. Many of them occur in Ecotaxa, but not all of them. And a reef is working along those lines to be able to upload these type of images to Ecotaxa to help improve the ability of Ecotaxa to provide an identification of the community of phytoplankton that are present. So this is just a brief summary of what the planktoscope is and what hydrocolor is to give you a sense of the type of information that can be collected. Now with the hydrocolor, it takes very little training in order to be able to collect data that are very useful for understanding the quality of the water. With planktoscope, it takes a little bit more training, but again, it does not take a high degree of training in order to be able to operate this and get extremely good quality data that helps you understand what is the composition of the phytoplankton community now. Does it contain harmful species, such as those in the Gambardiscus genus, or do you see changes in the phytoplankton over time? Now, you certainly would expect to see changes over seasons, but really what you're interested in is, as this climate change progresses, are you seeing shifts in the phytoplankton community composition that can help explain the changes in the fisheries, for example, that you may be seeing? All of this information helps you better understand your coastal waters. And when you better understand your coastal waters, you have a better ability to predict or perhaps help mitigate problems before they develop. So both Brin and ITI professors and scientists now have expertise with using the FishGIS, the hydrocolor, and planktoscope. And it's really time to put these tools to use. So I encourage all of you to, if you have interest in this, to get in touch with Professor Suhendahar or get in touch with the reef to figure out what you can do in order to be able to help collect more of these data. But observational data alone, we need those data, but it's not enough. How are you going to use those data then to help you make decisions that help you sustain your coastal waters and help the people that rely on those? And that's a brief introduction to our next talk by Professor Trick, Charlie, who is going to talk a little bit about that. So thank you very much again for the opportunity to speak with you. And it's a real pleasure to be here.
Okay, thank you very much, Professor. In this session, you have already explained us about FishGIS and a planktoscope for monitoring marine resources and environment. How these two innovative technologies are used to monitor marine resources and the condition of aquatic environment. Nice presentation. So the next session will be delivered So please type it. Type.
I think you can go ahead, Charlie.
Okay, I actually can't hear anything. Can anybody hear me? Mark, can you hear me?
I can hear
Yes,
you.
your voice is clear, bro.
Can hear you.
Okay. All right, thank you very much. I would like to be able to share my screen. I don't know. I think I'm gonna. Maybe not. I'm sorry, we don't commonly use Zoom.
Check in the center. There is a center and a middle. There is a green color there. Says
There we
share.
go. This is going to be confusing. I apologize for this. I do see the green
Don't
share.
worry.
I promise to make sure that.
Here you are.
Oh, can you see anything now?
Yes.
Yes.
Clear.
Does this look familiar?
Yes, clear.
All right. So we have a picture and you can hear me, right?
Yes.
Yes. All right. I have success at this late date. Folks, I am really pleased to be able to share my thoughts with you today and to kind of summarize ideas that the PICES, the MAF group, the FishPhyto group have kind of brought or shared with our friends here. What I really would like to do is take a kind of a social aspect and talk about you as a scientist and the communities that you wish to serve. So we're not going to be talking about phytoplankton, but we're going to be talking about why did the MAF group, the PICES group, invest in providing information, technology, techniques to our community here in Indonesia. And then I'm going to ask you to think about what responsibilities come with gathering this information. So if there's kind of four technical innovations that have occurred that are thought to change how we measure marine resources. One is to try to make sure we have as much remote measurements as possible, and we might use satellites, we might use GIS, or we might use many of the techniques that we have provided to Indonesia here, such as FishGIS. The second is not only should it be remote, where communities could probably assist, but make sure that there are new sensors available as well. These new sensors really could be such things as the microscopes that Dr. Wells just talked about, or the microscope analysis that Arif just talked about. But they're really information possibilities for sensors to be in the hands of both researchers and community members. Now, the third point is to make sure that this information is then put into a data system where data analysis or management could be possible. Opportunities to provide good data that people can share, people can analyze, both providing it to the community and providing a time frame on this. This is where this word sustainability will come in. We take a picture of our environment, we use the techniques, but we should know, if possible, what did it look like, or what will it look like if we continue to do that? So that's part of the sustainability story. But the most important thing, I think, is a purposeful systems analysis. Why are we taking these data? How does it fit into our decision-making, or our policies or our perspective of how institutes in Indonesia can actually utilize the information to provide leadership, provide innovation. Okay, I don't know who's controlling my screen, but there's the next one. There we go. Thank you. All right, so these are my personal thoughts. And again, if the individuals in the room here don't do this, then it will never get done. We have not a track record, but we have experiences to know that often we will provide technologies, techniques, ideas that just once we kind of go into the background, they don't go anywhere. So this is the time where there's this transition between what we can offer you and what we and you can work together on to both work with communities and sustainabilities. So the first thing is all these techniques provide data, but data without a perspective of how to use it is just a number. We can be happy with the number, we can do statistics with the number, but we have to have the perspective of what that number means, both in the past and in the future. And the other is that some of these techniques are not numbers, they're observations. Arif points out this beautiful community structure, and then Mark looks at the use of planktoscopes to understand the community structure as well. But if we just look at the community structures without understanding how they put into our perspective, they're really just lost memories. We're happy that the data is good, but we have to do something with it. Okay, so related to this, these are the kind of the first 2 points, but related to this is that in the room here are individuals that provide the organizational structure, the leadership to be able to stand up and talk about what is the relationship between the observations and the values of Indonesian science. Not just science, but the values. Are they answering questions? Are they providing innovations? And thus, are they valuable to continue on? So all the data can't just be the data that we have suggested are valuable, but rather they're the data that provides value to the purpose of the Indonesian sciences. So this value-based outcome is a great theme here. What I really like now somebody did within the group that's in this room here is to say, okay, I'm going to look at systems thinking. I'm going to look at how to model this material because these numbers mean something. They may mean something with regards to climate change. They may mean something about how we overfish or how there are fish that are kind of more usable for the Indonesian communities. They may say something about the energetics of the fish population when you use fish GIS, for example. Are they small fish? Are they large fish? Are they lower on the food chain? Are they higher on the food chain? These are important components to make sure that what we are measuring is something of value to the scientific community and thus leadership in making decisions. All right, now this is actually quite difficult because we have to put in perspective the organizational structure based on values such as, are we protecting or concerning local communities? Are we able to provide them information about how to harvest fish? But what happens if the fish are disappearing and there is no solution? Are we integrated into the economics of the country to be able to say that we would like to keep this community viable, but we recognize that fisheries are not part of it. So that's part of this countrywide economic planning. The data on the fish, the data that you collect on the quality of the fish or the levels of toxins, all have an impact on the economics of the fisher, economics of that community which they're at, and more broadly, the economics of the country. They also are related to seafood security, which is globally one of the most important things to worry about. Is there enough fish both to sell on the open market and to feed the masses, to feed the individuals within Indonesia? Or are we losing fish so that we are generally losing that ability to have a very good food source? Finally, as we have seen in our studies here, there is a recreational aspect of both fisheries, water quality, and kind of the coral reef strategies, because you can have recreational and tourist economies. The data can be used to sort out, do these economies of trying to improve the coral reefs, do they really improve the lives of Indonesians? Or is there a trade-off? So I'm trying to get the individuals in the room to say, yes, I'm going to hold on to this data. I'm going to have this technology. I'm not going to leave it on the shelf. I'm going to get my team to work together to utilize this data in a very logical process, using systems thinking, using a creative opportunity to understand that this is a time frame of how this information can be passed along to make big decisions. So the big decisions is now to move this along by having engineers or technical developers, data scientists, systems thinking, community members, all integrating together to utilize these values, this data, this information. So we can go to the next slide. There we go. All right, now here's a big issue. We talked a lot about communities here. So let's think about what a community might mean. Most of us think about a community that is something that is outside where we live. Okay, communities are elsewhere. Okay, and thus we would like that community to build on the gathering information for itself. And this is good. This is part of the early strategy, is get as much information as possible. But the community values might be different than the values that you might have in the life that you live. So it's really important to have this strong connection with communities in a variety of ways. First of all, we talk about community sciences, which are there collecting the data. But this is actually very challenging, is how do you keep a community actively involved in collecting data that is valuable to you, that you might want to present back to them as a good economic model? You have to step into that community. You cannot just visit that community. You have to be integrated into that community for this to have some sort of long-term success. All right. And by the way, when you do that, you're making a promise to that community. You're asking them to trust you, that you will come back and you will tell them that information about why the data they collected was important. Many scientists in this world collect data and they walk away. And I'm encouraging you all to use the tools that is related to PICES to build a relationship with a community of which you become invested in that community. Don't walk away. You have a promise to them. To be successful, you have to find somebody in that community that is a natural leader, because if that changes a lot, you don't learn very much. Okay. And there's a couple other components. Share the values that you have with that community, make sure they match. Have a good communication plan with that community. Don't just show up and then periodically show up. Let them know what the data means and why. Address privacy issues. I know that's like FishGIS has some privacy issues that it has treated, and I think is a very good model. And then eventually allow that community to govern its own data. If you challenge these steps, or if you accept these challenges within the steps, I think you will be able to recognize that you were doing this properly. So I think, is there one more slide? Yeah, let me just quickly point this out. You have innovation, you have smartphones, work on the technology of building smartphones, skill the technology. In other words, if you have individuals that are capable of modifying the data that's coming in, make sure that you develop those individuals to be good thinkers about what data means, just not good technical individuals. And again, the big part is try to communicate sustainability to that community or to the country in general. So that will improve resource use, improve food security, traditional life, and public health. So with that, I'd like to thank you for the time. I hope you can take this information and make sure that you can be innovative, you can be progressive, and you can be a great great example of how technology can lead to good discovery. So thank you.
Thank you very much, Professor. This is very, very nice presentation. And the last session today will be delivered by Professor Suhendar from BRIN, from Agro-Industrial Technology Institute Teknologi Indonesia, with the title PICES, Corporation on Marine Science for Human Resources and Capacity Building Development. 20 minutes time is with Prof. Suhendar, please.
Okay, thank you, Dr. Abu Amar. Can you hear my voice?
Clear.
Okay. Good afternoon, everyone. So here is I'm very happy to have opportunity to share with all of you about our activity in of the cooperation between PICES and the Indonesian scientists in. We already on this opportunity we would like to thank to Professor Sofiano, Rector ITI, Dr. Aryo from the head of Research Center for Environment, BRIN, Research Center for Environmental and Clean Technology, BRIN, Professor Jain also from ISO and Research Center for Oceanography for giving the lecture, and also Professor Makimo from PICES Tokyo University, and a flavor of the Professor General about Thailand, yeah, small island and research development. And Professor Makino talking about the how the project PICES activity in Indonesia. And also we already hear from Dr. Sean Timore talking about smartphone using PCIS, and Professor and Pak Arif from Paris University of I'm talking about the Gambiria's question about Ciguatera, and Professor Max about how the plankton spot can be very useful methodology to identify a huge data sample of plankton. And Professor Charlie Trick, about how the PCIS and smartphone and the other technology for monitoring marine resources is very important. So on this opportunity, we would like to present about, okay. So we would like to present about the PICES Indonesia Cooperation on Marine Science for Human Resources and Capacity Building Development. So my name is Suendari Sahuma from the Center for Environmental and Technology, National Research and Innovation Agency, BRIN, and also Department of Agriculture Technology, Institute of Technology of Indonesia, and Expert Council of Indonesian Research Association. Nggak bisa di ini. So we would like to talk in the first slide. Why is the PICES Indonesia collaboration is important for us? Maybe Professor Makino already introduced what is the PICES. The PICES is the North Pacific Marine Science Organization. It's an intergovernmental scientific organization that is established and held in the first meeting in its first members are Canada, Japan, Republic of China, Republic of Korea, the Russian Federation, and the United States of America. So the purpose of the organization is to promote and coordinate marine research in the Northern Pacific and Junction System. And also advance scientific knowledge about the ocean environment, global weather and climate change, living resources and their ecosystem, and their impacts of human activities. And also promote the collection of rapid exchange of scientific information. So while in Indonesia is the large archipelago country in the world, and with it is with marine area, shelf area, coastline, and population almost 70% living in the coastal area. So it is very important for us to transfer knowledge from the PICES countries in the North Pacific, in the subtropic area to Indonesia as the largest equivalent country in the tropical area. And if we look the activity of the PICES here, so it's a very wide scientific area can be adopted in to manage the coastal marine resources in Indonesia. So we look here, there is a marine ecosystem, human system, processes, and climate. So it is all scientific subject is can be transferred to Indonesian scientists and all policymaker and also government to manage research in the offshore area or coastal area with reach of the coastal marine resources like coral reef, seagrass, and mangrove. all these resources can develop to improve the well-being of the coastal communities with the many activities like brackish water, tourism, agriculture, yeah, aquaculture, and so on by a sustainable manner. Hopefully, in the future, our marine resources can develop for our community well-being can be sustained. So this activity also PICES with the 6 countries member can we collaborate with our Indonesian scientists from not limited on the right now ITI, PRIN, and Mataram University, and also Nusa Tenggara University and Pajajaran University on BKPN. But also another we open to another institution for the collaboration. Here with I inform you that the PICES Indonesia collaboration activities as presented by Professor Makino before is have a long stories from 2007 I heard and with our Indonesian country also we have developed from year 2012 to 27 and marine ecosystem health and autonomy. Also in this term we introduce this autonomy concept to marine ecosystem health and human well-being as a concept to develop how sustainable fisheries and coastal management in Indonesia. And so we did workshop more than 100% training and 5% in Jakarta, Karawang, yeah, in 90 and 2030, and in Pekalongan 2004, and in Jakarta, Muara Gembong, and Remayu for field trip. And then in 2017 to 2000, so we have a program of the PCIS. Is already done, already delivered by previous speakers. This program is the activities for building capacity for coastal monitoring by local small-scale fisheries. Also, we already did workshop and training in Serang, Banten, and in Jakarta, Seribu Alian workshop, and Serang, Banten, and Pelabuhan Ratu. And then in 2023, we have a program of Sigatera on building local warning network for the detection and human dimension of Sigatera fish poisoning in Indonesian communities. And also we did a Sigatera research survey in Limata, Lombok in 2022 and workshop more than 30% in Lombok in January 22. And now we have a program from 2020 23 to 26 by program FishPhyto, how to create a phytoplankton fishery resolving program for sustaining local communities in Indonesian coastal water. And right now we have a general lecture in instead I think we already record there is more than 100 person, yeah, in the sport and in concern, and also we did a research survey in Limata 2024, and general lecture is now in 2025. So the upper goal of the project is to provide local communities operational capacity and knowledge to manage fisheries resources and ensure sustainable seafood safety in Indonesia. I think these topics already presented by previous speakers, yeah, in keynote and also in scientific technological matter, yeah. and also how to identify research need for deploying this tool on PICES member countries. So I hope everybody, if we can discuss later if there is something to us. Here is the area where we have disseminate the PICES technology in Jakarta, in Jakarta, Pekalongan, Gili-Matra, and also we already introduced to Bantaing, South Sulawesi, and also in Raja Ampat, yeah, Raja Ampat here, and also in Seribu Island, Banten, Ancol, yeah, Cilincing, and so on, yeah, Damai. Here is the implemented agenda for 2026. Yeah. There is a quite activity in 2002. Yeah, in May and October, December and February. It is almost cover all season Indonesia for study of Cibatera and Ampulan. It is funding by PICES and also RIM auto Cyrus, like this one. Also, we did workshop and training in January 22, 27, 20, yeah, and Sigatari survey on 22nd. So we still have activity on this year by RIM project of BRIN for research for advanced Indonesian. And 2020 this year we have two survey again for transition period in June June or November. So how to deliver and disseminate the technology? We try to develop the model about how the strategic planning and implementation PICES Indonesian collaboration. Like here we look in the right side, there is a CFP, Sired, supporting by Sired also, Small Island Research and Development, CFP. There is PICES with all member from the 6 country here, from and various institution. There is an Japan Research Agency, Hokkaido University, and also School of Mine and University of Mine Technology and University of London, Asia, and so on. And also we collaborate with GSPS, CREPSAM, and LAMER program, and so it is all how to encourage our scientists to do and policymaker to have activity in Indonesian research. And so we involve coastal community of Gili-Matra, like for example, and in other place in Seribu Island and so on, yeah, and who involves on the workshop and training. And so there is a supervisor group society, for example, in tourism area in Lombok, and other class bottom board and small hotel association and so on. So it is all stakeholder from the community and who involved in this activity. And what is kind of the dissemination is a workshop training and a general lecture like right now. So another institution is Indonesia Institute Technology Indonesia, University Mataram Indonesia, University Pajajaran, and others. So we still open for the other institution. What is the activity is data collection, research, and Pangtoscope, like Pangtoscope, Hydrocolor, PCS, and also we involve on the researchers, student, and also practitioners. Practitioner, it's mean from the the community who who need who support for their activity. Also in the high level government, we coordinate and involve with the Ministry of Environment, Ministry of Fisheries and Marine Bio, so Ministry of Tourism and Creative Tourism, and Regional Research Innovation Agency and European National Master. So it is a scheme of the bottom-up and top-down approach strategic model to disseminate oxygen therapies positioning and by involving various stakeholders in local, national, international level to local stability of Golimata. This is the model, and this model can apply for the another project and activity. So here is, I think it is already introduced by Professor Martino before. Where is this model, bottom-up model, and funding source to meet to support the activity for capacity building, workshop planning, and also national research, local government research institution, and also for policymaker and data collection for research to develop sustainable marine coastal resources and human well-being. Here is one of the research and innovation funding brin. I think we call is RIM. So we include the technology from PICES to workshop training like Parkinson Code, PCIS, hydrocolor and also we introduce to the Belitung if it is possible. Here is the activity, the first activity of the workshop and training in Jakarta, I think, and also in Karawang. And we built a survey how to introduce the Statu Umi for marine ecosystem health program of the PICES. This Satu Ilmu program is how the aquaculture can provide sustainable technology by the introduce the EMTA. EMTA is integrated aquaculture multi-product aquaculture. So by EMTA technology, the aquaculture technology to be zero waste because all all commodities have an ecological and economic value. Ecological is how to seaweed can reduce the unorganic matter and oyster or benthic organism can be reduce the organic waste. So there is four component like seaweed, shrimp, and also triposom, and green mussel we use in here. See, for how to increase the diversity, also we introduce the culture of the soft shell in Karawang. It is also we introduce how to plantation the mangrove here, introduce in cooperation with the Ministry of Fisheries and Marine Affairs. This is one of the activity of workshop training also here in Karawang. In West Java, here is the second workshop in Kalongan. With Kalongan also we introduce how the human marine environment health by using Satu Umi concept can be introduced to sustain the coastal marine resources here. And we how to import the rob problem. How to solve the rob problem in Kalongan for the other activity like fisheries and so on, and also for sustainable of shrimp culture here. and also how to restore the mangrove, yeah, to sustain the coastal marine resources in Kalungan. And yeah, with sustainable multi-tropical aquaculture here. And also in Seribu Island, we have a training and Satu Umi in for marine coastal environment health. And in Bantaeng, here is in Bantaeng, how to introduce the PICES technology in Bantaeng with seaweed, it's seaweed. And then it is how the lecture to transport ni yang lagi Professor yang lagi from the PICES also. Yeah, and it is from yeah. And the others is in Israel we have a signing ceremony before in 27, yeah, with PICES and to develop a space GIS here. Also here is we give a training program to Muara Gembong people, coastal community, how to use the plants, not plant scope, but it is fault scope, yeah, fault scope. Fault scope is a kind of mobile microscope for identify plankton and here by using smartphone as well. This picture also introduced by Professor Mark. Here is the activity with the Muara Gimbong coastal community to adopt this smartphone utilization for PICES and hydrocolor. Hydrocolor already introduced by for Professor Charlie for water quality monitoring using a smartphone. It is very useful for coastal community to help the policymaker to report the status of the water quality in the coastal area. This practical use of the smartphone in the Muara Gembong introduced by Professor Mark here, with PICES team in the field. There is probably Professor Charlie and so on. Okay. And also here in Pulau Seribu, we introduce how to use the smartphone for water quality monitoring in Indonesian coastal area. Same it is in the Serang and Banten and also in Pelabuhan Ratu. Here is in Pelabuhan Ratu. And here is in 2022 and 2024 we did a survey of Sugatera. It is how to develop mitigation model for Sugatera and HAB in Gilimatra, Lombok area, in Lombok. And it is the workshop and training in Lombok in January 2023. So we audience with the secretary, regional secretary and governor of the Western Nusa Tenggara in Lombok to do. Here is the community of the Gilimatra who the PICES program have giving training how to use the smartphone for using for monitoring marine environment, also identify phytoplankton using plankton scope here. There is Professor Mark here how to just to do coastal management and train giving training in West Nusa Tenggara. for the Gili Matra from Avasta community. Here is the agenda for the training. I hopefully all Avasta are already involved on this matter. It is how to introduce the file system, the field area of the Gili Matra here. It is a very interesting area for tourism destination, and it is need attention for maybe global scientists how to sustain this very important destination tourism area. It is also safe with workshop and training in July in Gili Matra. Okay, maybe that is introduce the activity how to build human resources capacity building and how the importance is the cooperation between PICES and the Indonesia. As summary, we brief here that the PICES in this collaboration is very important and very useful for transfer of knowledge and technology in the marine sector from developed country to developed countries to improve the ability of the scientists and community to understand the importance of marine resources for sustainable development and improve the welfare of coastal marine communities in Indonesia. And general lecture and scientifics also are very useful for exchanging knowledge and experience in various research activities, and their application communities need to be continuously developed and to implemented to facilitate scientists to transfer their knowledge for human resources capacity building. And dissemination of science, technology through workshop and training to the community are important and necessary to provide and increase their knowledge, understand, accept, adopt, and apply in various related activities. And a bottom-up and top-down approach involving community leaders from price-related groups as well as decision-makers of both the local and national levels, including experts from various research institutions, universities, NGOs, and a strategic approach to be implemented in Indonesia and might be in other countries globally. To socialize model to bridge the application of science and technology in the field of marine science to the community, and governance needs to be continuously developed, refined, and regularly and systematically so the implementation is application in the field of science. Okay. So it is next. Thank you for your kind attention.
Thank you very much, Prof. Suhendar, for a nice presentation. Now, ladies and gentlemen, so we can discuss in this session. Please raise hand if you have questions from the floor and who your question is addressed to. Yeah, Professor Hen, and then please mention your name and address to.
Baik, thank you, Prof. Mr. Abu Umar, and thank you for your presentation today, Professor Suhendar or Mr. Suhendar. Let me talking about your presentation today. The first time I would like to say it is very, very important. I'm sorry. It is very, very challenging to make the other research about your material today. Especially, I hear your presentation about the some research, especially in Indonesian. All right, Mister? Yes.
Yes, yes, yes.
So, you know, there are many, many research in Indonesian which is completed with the many, many institutions. For example, maybe from the higher education, brain, so on. But I think you should know there are many, many research of the research is not implemented in the reality, right?
Yes, yes. Mm-hmm.
So, can you tell me, how do you think about the research if the one research is completed by the researcher, maybe by Professor Sohendar, maybe by the other researcher. But you know, in Indonesian, the results hasil, the research is not implemented in Indonesian. So can you explain to me, how do you think about it? And how do you think about your explanation, solution about this? The first question. The second, I hope you can clear about my question. Maybe Mr. Abu Umar help me to make it my question to Professor Sohendar, okay?
Okay, thank you. That is enough for me. Okay.
Okay, the second.
Okay.
Thank you
Maybe
very
just
much.
only one question. Maybe
Okay,
just
okay,
only
thank
one
you.
question
Okay,
because
okay, okay,
yeah,
thank you,
okay.
thank you. I'm so sorry. Thank you,
Okay.
thank you.
Yeah.
Okay, thank you very much for a very good question, Professor Hen. Yeah, Professor Hen. I think we already, for experience, we already introduced. Maybe later Professor Max and Professor Charlie, yeah, and Dr. Sion can be added to this. Our experience with introducing of the PICES program in Indonesia, of course, but not yet implemented widely, but locally we already implement. The first one, how to introduce the marine environment health for food safety with introducing the Sato Umi, Sato Umi program, Sato Umi as a concept of model. I think we already look in the Pekalongan and Karawang. They already implemented how to apply the integrated multi-tropic aquaculture to reduce the waste. Also, how to increase the biodiversity in. I think we look in the Raja Ampat. The last, there is how to the plantation of the replantation of the coral reef can reach the biodiversity, something that, and also how they use the smartphone to identify the marine environment and also identify the fish resources, coastal environment, also water quality monitoring by pricing. But if we would like to more wide apply, so the government should be adopted and should be have a program. For example, for regular monitoring by involving of the coastal community. If the government have a program regularly for monitoring with involve of the coastal monitoring that we already train and produce, I think the technology that adopt from the PICES and from another technology, another source maybe can be applied widely. So so it is, I think, the supporting of the government in the high level and local level, and also as scientists, as mediator, maybe, how to how to link the scientist, the scientific from the scientist to the policymaker and the community. Maybe it will be more effective. And will more widely. So it is challenging for us, I think, because without involving of the government, I think because there is no sources and funding and something. Maybe you understand this question like that. If we have found the scheme, another scheme active for funding activity, for funding resources and something for the activity, I think we will very useful. So the concept of the not scientific for not scientific for science, science for science, but science for the how to develop the economical and so on. Maybe we can we can run well. I studied. Maybe Professor Mark, Professor Sali, maybe Professor Jainal, yeah, maybe uh, and Dr. Sion, maybe Pak Arif, maybe can we add? Thank you.
Please.
I don't have anything or much more to add. I think that Professor Suhana has sort of given a good overview of what the ideas are. This project obviously couldn't cover all of Indonesia, so we had to pick certain communities to try and work there to show sort of the strategy for how to do the capacity building, and what's necessary now, ideally, as Charlie has mentioned, is now to start trying to expand it to other communities. And not being from Indonesia, I don't really know the linkage of how best to do that, but I think Suhenda has presented one strategy to go forward that could be well successful.
Thank you. Maybe Professor Charlie, will you add?
Yeah, maybe I'll make a small comment here, and that is we are globally far away from doing science and marine resources just for the sake of doing science. I think we depend upon the marine resources to provide security for our countries. And while everything we have done here provides some tools, Professor Sahindar has pointed out the fact that it really is for the value of Indonesia, not just for scientists in Indonesia.
Okay, we would like to continue the question from the audience. We have already compiled. There are 6 questions from the audience. The first question is from Muhammad Iqbal Habibi from BRIN. The question is, how do you detect the harmful alga bloom and Shuketera fish poisoning in marine environmental? What is the parameter between them and what the difference is? Maybe from Professor Mark or Mr. Shian Takimura or Pak Arif, or maybe for Professor Charlie, please.
I would suggest that Arif is probably the best person to address that question.
Okay.
Okay, thank you very much for this and for the question. So how do you actually detect the harmful algal blooms and etc.? One of the way we already talking here is about we are doing the sampling. We are collecting the sample in the water column if we are looking at the planktonic harmful algal blooms, or we are collecting the benthic samples that we scrap the macroalgae, we collect macroalgae, dead corals, and other substrates if you wanted to see the benthic harmful dinoflagellate. The best way to do it is collecting samples and analyzing it. We can use the traditional method like under the microscope, or like Professor Mark already explained here using the microscope. There's other tools that are available. We can actually try to detect the presence of the toxins, but it's quite complicated. We try to do it with passive sensor that we co-develop here in France and some other countries. We try to develop the passive sensors specific to several toxins, but it's a very complicated process that unfortunately for Indonesia right now, we don't have the capacity, especially to detect the Sigua toxin. But Indonesia now has ability to detect other toxins like the amnesic surface poisoning and domoic acid that causing amnesic surface poisoning and paralytic surface poisoning. The Bureau of Quarantine in Ministry of Fisheries, they are able to detect and give you the concentration of these two particular toxins. But unfortunately for Ciguatera, even globally, it's still very hard to analyze that. And we don't have the rapid kits to detect Ciguatera toxin in the environment. With harmful algal blooms, you can look at the waters, collect samples, or sometimes we use the satellite to detect the change in the presence of the increasing chlorophyll. But yes, to know what species that are blooming, you have to go to the field, collect the sample. This is now the way for now.
Okay, Pak Arif, thank you very much. The next question from Agus Budi Purnomo, PT Amitas. That is a general question. How to preserve marine ecosystems and maintain beach abrasion? Professor Charlie or Professor Mark or maybe Professor Sian Takemura, please. This general question.
So I'm sorry, my connection broke up a little bit. What, can you repeat that question? I missed the last part.
How to preserve marine ecosystems and maintain beach abrasion?
So the maintaining of the ecosystems is basically you have to understand. I mean, this part of the whole project is to provide tools to make measurements that help you understand the state of your local system. And so if you want to try and preserve it, you need to be able to understand whether it is changing, or if it is changing, how it is changing, and whether that affects the sustainability of that system for whatever you may be using it for. For the beach erosion, that's something we didn't actually deal with in our project, but Suhendra-san, you mentioned, and you could talk about the mangrove restoration project from the beach erosion perspective.
Yeah. Yeah.
Okay, maybe
Yeah. Yeah. Yeah, I think what is the mention by Professor Mark is already clear. We have to understand the marine ecosystem situation, and we have to understand that the marine ecosystem is polluted or not. And of course, we have to understand what is the technology for detect of the polluted marine environment. I think the hydrocolor technology by using a smartphone, as introduced maybe Professor Max or Xion, and yeah, and also Pak Arif and Professor Charlie, it is very useful because with smartphone with hydrocolor we can detect at least the most important parameter like turbidity and turbidity maybe. It is the important thing. What is the problem turbidity due to the erosion or sedimentation and for silt or from plankton bloom, something that. If we already understand about that one, we can record by the smartphone by using hydrocolor. Hydrocolor also we can analyze chlorophyll A as a water quality indicator. So we will understand how much the polluted level of the marine ecosystem, so we can report to the government, to local government, on policy maker what is the situation of the marine ecosystem or water quality in the near where is the you found that. Yeah. As for episode, yes, yes, I think already clear of the Professor Marx. Mangrove plantation is very useful to protect the erosion in the beach. Yeah, by maybe everybody already know the function of the mangrove forest. to protect the coastal area from the abrasion from the wave and something like that. Yeah, yeah, yeah.
Okay, thank you very much, Prof. Suhendra. Effective is FishPhyto technology, this marine environmental monitoring in detecting and mitigating potential harmful algal bloom outbreaks in coastal and small island region of Indonesia.
Professor
Sorry, there
Max,
was
maybe.
heavy echo. There was a heavy echo there, so I didn't get the full question. Could you repeat it again, please?
Okay, how effective is FishPhyto technology, this marine environmental monitoring in detecting and mitigating potential harmful algal bloom outbreaks in coastal and small island region of Indonesia?
Okay, as far as how effective is, say, planktoscope and these approaches for measuring harmful algal blooms, they can be very effective if they're applied. As Arif-san brought up, we need the samples in order to be able to understand what's going on. We cannot tell from satellite images whether a harmful species is occurring there or not. They're trying to develop new satellites, they're trying to develop techniques to help with that, but they're not there right now. So in order for these methods to be effective, we need to be able to have people collecting the water samples and then running those samples through, say, for example, the planktoscope in order to gather those data. If you're doing that, it can be very effective. So in the United States, on the coast of Maine, for example, they have many people out right now that will collect samples, coastal waters, and they look under regular microscopes. They haven't implemented planktoscopes yet. And they're citizen scientists. They're people that are volunteering to do this. And because they have so many people doing this, they have a very effective way of monitoring for the harmful algal blooms. But it really depends upon people stepping up and deciding that they wish to know whether this is becoming a problem so that they can help provide the information to Arif-san and others that will be able to then take steps to try and mitigate whatever those blooms may be causing. And mitigation can take different forms. So if you start to see with time, for example, that your chlorophyll concentrations are building up over a period of years, that may be due to more runoff bringing nutrients in. Well, you know that eventually that's going to generate problems, such as you have in Jakarta Bay. And so if you start to see that early on, it may be helpful then to try and mitigate by considering how can you reduce the nutrient inflows so the problem, you basically mitigate or avoid that problem from developing. But it all comes down to having people being able to collect the samples, collect the data that's necessary to try and interpret that.
Thank you very much. The last question.
Can I add a small thing here? Yeah,
Okay,
and
please.
that is everything that Professor Wells talked about requires an understanding of time. What did it look like? What does it look like now? And how are we going to continue this? And that takes an investment in a community of individuals willing to look at this. So I think it's one of those things that if this is important, you put the energy into doing this correctly.
Okay, the last question from Chandrika Ekalarasati, University Mataram, Lombok. Can this FishPhyto method also be applied to mitigate eutrophication cases in Bima Bay in Lombok? Can this FishPhyto method also be applied to mitigate eutrophication cases in Bima Bay, Lombok?
Well, these tools can provide information of whether you have problematic eutrophication. The tools themselves aren't going to solve that. It will require the community to recognize that this eutrophication is becoming a problem. And the tools can provide that information for the community, but in solving the problem, it will be up to, as Charlie has pointed out, it's up to the community to use this information to then make decisions about how important is it to try and reduce, say, the eutrophication, and how might you best go about doing that. And so the tools just let you know the state of the system and whether that system, if you're doing this with time, whether that state of the system is changing. But as Charlie points out, it really is going to be up to the community then to organize around that information to figure out what decisions should be made. Charlie, you're better at talking about those things than I am. So
I don't think I can add anything that would provide more flavor than what you just offered. So well done, Professor Wells.
We would like to thank all the speakers, all the audience. Here to me. Okay, because this time also has to end this session. We would like to thank you all the speaker, all the audience for the participation in this event and see you the next event. So we give time to
okay, well, thank you very much for the opportunity to participate in this.
It was a wonderful
Welcome, Professor
Thank you
Mark,
very much,
Mark
everyone.
Sang. Thank you very much, Mark Sang, Charlie Sang,
Okay.
Sion Sang, Pak Arif Sang, from Paris. So maybe all night and
all
now
the sounds.
Yeah, I think
midnight.
colleague in
you very much.
North America is
Anyway,
really late night.
yeah, really late night. Thanks so much for your time, for your time. Yeah, maybe
Okay.
we have to hear the closing. Yeah, 1 minute. Yes. Yo, maybe langsung Bu Santi aja, Bu Cinta aja. Maybe Cinta Sang will close the session with 1 minute. Ya, ayo, udah. Ya, ya. Ya, ya. Udah langsung aja Bu Cinta.
Oke, baik. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. Greetings to all of us. We from the Department of Agro-Industrial Technology, ITI, would like to thank you so much. This general lecture and scientific meeting has been well carried out. We would like to express our gratitude thanks to Rector of ITI and all IT management and academics. Thanks to head of the Center for Environmental Research and Clean Technology of BRIN and his staff. Thanks to the delegation of the North Pacific Marine Science Organization, PICES. Thanks to all speakers and moderator, as well as all the participants and invitees who have attended this event. Ladies and gentlemen, allow us on behalf of the organizing committee to report the following. The purpose of the general lecture organized by ITI and PICES, supported by the National Research and Innovation Agency, BRIN, and various other stakeholders, to increase knowledge and increase the capacity of human resources in the field of marine science and technology for monitoring aquatic environmental resources in coastal areas and small islands. Approximately 100 participants and invitees attended this general lecture from PICES, ITI, BRIN, and other government levels in the Tangerang region and Banten province. In this general lecture, several material related to FishPhyto and marine environmental monitoring for disaster mitigation of harmful algal blooms and Chiguatera fish poisoning and various phenomena in the field of marine science that are currently to be global uses have been presented and discussed. On behalf of the committee, we would like to thank the expert from PICES and all participants who have been willing to attend and contribute their knowledge in this general lecture. Hopefully, this activity can provide benefits and can contribute to the development of science and technology of marine science globally in the international world, including in Indonesia, and can be used in the development and utilization of natural resources, including agro-industrial technology, food safety, and others to support the acceleration of national development and answer global challenges through technological development and improving the capabilities of human resources. By saying Alhamdulillahirabbil alamin, we officially close this general lecture and scientific meeting event. Goodbye and thanks. See you next time. Wassalamualaikum warahmatullahi wabarakatuh.
Yeah. Thank you
Thank
very
you,
much.
everyone.
Okay, thank you very much. See you. Bye.
Thank you very
Bye.
much.
Yeah,
Bye.
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 Bye. Bye. Bye. Bye.
Thank you,
Yeah.
thank you, thank
Thank
you.
you. Thank you.
Bye.
Thank you very much. Bye.
Thank you very much, Mr. Agas. See you next time.
Alhamdulillah. Terima kasih Bapak Abu semuanya. Ya.
Ada soal